Hello, everyone. What we're going to discuss in this lecture now is the experiments that were done by this scientist slash monk, Gregory Mendel, which have led to the birth, if you want, of genetics, how we understand it now, or at least partly how we understand it now. You'll see at the end of the lecture, there are many exceptions to the genetics we're going to describe here. So going through the outline here. So first we'll talk about Mendel's experiments. What type of experiment he did with the pea plants, what are monohybrid, dihybrid crosses that he did. And obviously we will focus on the interpretation. What was the meaning of these experiments ultimately? And then we'll see what was the math behind the interpretation that Mendel's came out with. Why his interpretation are the only way to explain his experiments. Okay, and then in the end, we'll finish it up talking about the many exceptions. So Mendel's has helped starting genetics, but in the end, you'll see there are many exceptions to the genetic that he has described, which again, have enriched the understanding of genetic transmissions of characters. So starting with Mendel, you have to understand before we talk about his experiments, where we were at before he started doing his experiment in the second half of the 1800s. People did not really know how characters, and by character what I mean is specific characteristics that an individual have, like for example, hair color, color of the eyes, color of the skin, being tall, short. That's what we're talking about when we talk about characters. And by trait, what we mean is the possible variance of that character. For example, if you're talking about color of the eyes as a character, you could have brown eyes, blue eyes, green eyes. These will be the different traits. Same thing for plants. If you're talking about the color of the flowers, that's the character, and the traits could be purple or white, something like that. So people did not really understand how these characters were transferred from one generation to the other one. So what was the genetics behind that? And the most accepted theory was the blending theory, whereby people thought that, okay, in the next generation, what you get is basically a mix of what the parents had. So if you are a plant, the color of the flowers in the next generation will be a mix of what was before. But there were evidence that would support that and that would go against that. For example, if you look at the evidence supporting that, look, for example, at the kids generated by, for example, an African-American person and a Caucasian person, they would tend to have, for the color of the skin, something intermediate, right? So that would support the blending theory. But then if you look at the color of the plants, you see that there is no real mixing. In most cases, there is no mixing in the flower color. It's not that if you mix together purple and white, you get a lighter purple, as you would expect from the blending theory. So there were people for this theory, people against that. And that's where Mendel came in. Mendel is, just giving you some background about him, is a monk. He had done studies in mathematics and statistics, which were the big important key factors in their interpretation you'll see later on of his data. So he was a monk. He had the chance to study as a monk. And so the important thing is that he was doing experiments that many other people were doing at this time. Many people were trying to understand the genetics behind the transfer of characters between different individuals. But the key things that helped him a lot was, as I said, his background in mathematics and statistics. He analyzed his data in a very, very solid way. Not only that, but also he found the perfect system to work with. He was very good at finding a system that would guarantee basically high throughput in terms of data, at the same time very easy to manipulate. And the one he studied was the pea plants. Okay, that was the system he worked with. It was pea plants, which allowed them to generate lots of data and very solid. So the first thing he did, first of all, whenever you start an experiment, if you want to have a good model, what you want to have is something that allows you to start with very, very pure starting point. So something that's very clear for you, very understood. You want to have, for example, you're studying a disease in a mouse. You want to have mice that you're guaranteed that don't have it, and mice that you know is going to get it. But you want to have pure lineage there. Okay, so mice that you know They're resistant to this disease, for example, and mice that you're guaranteed they're gonna get it. Not that sometimes they do get it, sometimes they don't get it. You wanna start with pure material. And that's what he did with his pea plants. So what he did, big thing was, he started with what is called true breeding plants. So plants that have been, by using self-pollination, so these plants can self-pollinate or cross-pollinate. What it means is that if you look at the flowers of these plants, they have both the female gonads, the carpels, and they have also the male anthers here, which these produce the pollen, which then ends up on the carpel, giving origin to the next plant. So now, since they have both, plants can actually transfer the pollen naturally from their own anthers to the carpel. This is referred to as self-pollination. This basically will allow you to start. So if you allow for plants only to self-pollinate, okay, not cross-pollinate, in the end, you'll come out with a generation that will guarantee you that will have their characters, their traits for a specific character. For example, the color of the flowers, you're guaranteed that always give you the constant color that you want. For example, you have a lineage that will always give you purple, meaning that the genetic behind that can only give you that. They're going to be pure for that specific trait. That's what you want to start with. Excellent starting point, right? The same thing, if instead of using purple plants, use white plants, now you can let them self-pollinate. You start with pure breeding, true breeding plants for the white color. So with the pure genetic for that specific trait. And then you have the advantage also that since you can actually cross-pollinate them too, so you can actually remove the anthers from a plant and basically allow for, you can take the pollen from one plant and transfer to another one. You can do it easily with a little brush. You can do that. So you have a system that allows you not only to generate pure breeding plants, but also to play with them, cross them at your own please under very controlled conditions. That's true science here. Not only that, but then these pea plants could generate so many organisms in a very short period of time. In a matter of a few days, you can actually have the next generation. All you have to do is take the peas that are produced by these plants following either self-pollination or cross-pollination, plant them, and then you get the next generation very quickly. So you have a lot of data. You can produce a lot of data in a short period of time. Excellent. And then together with that, Mendel also analyzed the number of plants with certain traits for specific characters in a mathematical and statistical way, which was extremely solid, allowing him to come out with specific conclusion. All right. So what did he study? Well, he studied different characters in these plants. So he studied actually 7 different characters, the shape of the seeds, so these plants can give origin to 2 types of seeds, either fully round and smooth or wrinkled. Study also the color of the seeds. They can be yellow or green and so on. So these are the 7 characters that he studied. What we'll focus on in his experiments is the ones done for the color of the flower. Okay, so but all his experiments led to the same conclusion for all of these characters. And then in a second round of studies, he actually started combining 2 characters at the time, doing what is called dihybrid crosses. So he studied all of them, and the good thing is that all of these data that he generated, so he confirmed his theory in all of this characters, using multiple characters, again, solid science behind it. So he studied all of them. The one that we focus on here is the color of the flower. So the first character, or one of the characters that he studied, and we move on now to the study here. So starting the first plants, first of all, he generated true breeding plants for purple and white color of the flowers. Okay, so again, it means that allows for some purple plants to self-pollinate over and over and over. So he was sure that at the end, he was starting with a generation of plants that were pure from a genetic point of view for whatever was carrying the trait purple. And the same thing he did it for the white plants. He let them self-pollinate over and over and over, so they would have been pure for whatever they were carrying. It was allowing them to be white. Okay, solid scientific start. This is what you would call the parental generation, or P generation here. Let me just highlight it here with the pen. So this is the P generation it started with. Solid generations with only one determinant for one of the two characters. And then after that, what he did, he let them cross-pollinate, sorry. So basically, all he did, he was taking some pollen from the purple plants, transfer them to the white plants, or vice versa, one or the other. And so what he did, he got what is called the first filial generation, or F1. And what he observed, forget about the letters here, this is already an explanation which can ignore for now. What he noticed is that now, if you cross pure purple plants with pure white plants, what you get, all plants are purple. Not only they're purple, but identical in the intensity of the color. So these plants here are identical in intensity to the purple plants that were part of the parental generation. These already excludes, if you think, the blending theory, right? If it was blending theory was true, you should have a lighter purple color, but you observe no. Not only that, so these, if you think, tells you that whatever determines the purple color is somehow masking, in this generation, whatever determines the white color. Okay, that's the concept of dominance. Whatever determines, we know now it's we call it allele, but whatever determines the purple color is dominant over the white. That's how we would say it now. Then, it didn't stop here on the first filial generation, but what it did was another amazing thing. It took these F1 plants and it allowed them to either self-pollinate, so now they should be all genetically identical because you started with pure populations here, so meaning that these should all be the same too. To either self-pollinate or cross-pollinate, which would be basically the same thing. And you looked at a second filial generation, which is what you call the F2 generation here. So these plants are either self-pollinating or cross-pollinating, doesn't change because they're all identical to each other. And to his surprise, he noticed this, which is a key, again, to his conclusion. He noticed that 3/4 of the plants were actually purple, right? But the white color appeared again. So the white color had not really, whatever was carrying the white color had not disappeared in the first filial generation and had reappeared back on the second filial generation, the F2 generation. Not only that, but that's a key thing. He actually counted the plants, the number of plants, and what he saw is that the ratio between the purple plants and the white plants generated in the second filial generation was a ratio of 3:1. So you had 3 purple plants for each white plant that was generated. This is key. Wow. So not only that, he quantified his data, excellent scientific approach, but also he repeated the same experiment with different characters to make sure that what he was observing was not just unique to the color of the flowers, but to other characters too. And what he saw, you can see here, that he was getting similar results with other characters too. He was getting a similar 3:1 ratio if he was studying the color of the flowers, but also if he was studying the shape of the seed, the color of the seed, the shape of the pod, and so on. All of these characters would give him basically a ratio more or less equal to 3:1. So based on this data, now he came out with a series of conclusions. I hope you can appreciate it's not just a single experiment. He did many experiments here, which were basically supporting his theory, which was the following. So which stands on 3 key things. First of all, what he said is that now each character, so the color of the flowers or the shape of the seed, it's carried by a pair of factors in each individual. So in each plant, you would have 2 factors. He didn't know there were alleles. He didn't know about genes. He didn't know anything about DNA at that time. All he knew is that there were 2 unknown factors in each plant that determine each character. Okay. And now, the other thing he came out with, and then we'll see the math later on behind this, is that in order to get those numbers, the 3:1 ratio at the end, these characters, so each individual has two two alleles, we call them alleles, two factors here for each character. Now, when they generate the gametes, only one of the two factors ends up in the gamete. We know that. That's what is happening. If you remember when we did meiosis, you get one chromosome for each part. So one allele in each gamete in the end for whatever gene it is. And then this segregation, the way they end up in one gamete or the other one, is independent for what they carry. So whether you carry purple or white, you will have equal amount there. So 50 and 50 of gametes would have one factor, 50% would have the other one. This is key to understand that. Okay, we'll see later on why when we talk about the math, why it cannot be different than that. And then what it says that now, even during fertilization, now the way these factors meet each other, so the gametes carrying them, it's independent from what they carry. Again, all of this, only through this you can explain a 3-to-1 ratio. I will see the math later. So now we know, talking about genetic as we know it now, we know that now when you talk about a character, you are talking about a gene that determines that character, which has two possible types, which are referred to as alleles. So the same gene exists in two different forms referred to as alleles. And then one allele ends up in the gametes. So you would have one allele for purple flowers, one for white flowers, and the combination, that's what determines what he saw. Another important, so first of all, each character is determined by two factors that do segregate independently from what they carry. So each one of them ends up in the gamete independently from what they carry. Second thing is also that one of these characters must be dominant over the other. And that's how you can explain what you see in the first filial generation. So that's the only way to explain how, if you cross purple plants with white plants, you get only purple plants. It means that one of these two factors, that allele that is responsible for purple, is dominant over the white one. So if you have a mix of them, purple will take over, right? So that's the concept of dominance there. And again, it's what we know is that the purple allele in this case is dominant over white. The same thing for all the other characters. And then, as we say before, so now the gametes will contain 50%. So the way in which these alleles, these factors segregates in the gametes is independent from what they carry. So meaning that if you look at your sperm or your eggs, the chance of having one allele versus the other one, if you have two different alleles for whatever gene you look at, is going to be 50-50. So 50% of your sperm will carry one, 50% the other one. It's independent from what they carry, okay? And that's important, again, to determine the 3-to-1 ratio. And then when you have fertilization happening, also the fusion of these alleles, or the gametes, the chance of fusing with different type of gametes, so the chance of one sperm to meet a specific egg, it's independent from what they carry. Okay, and that's the principle of independent assortment there. And that's the only way, again, we'll see the math later on, to explain what's going on, the 3-to-1 ratio. If you change anything, so this is the independent assortment there, the idea. So all of this, it has to be 50-50 in order to get a 3-to-1. We'll do some example later on. We'll go through some math there. Okay, so the experiment of Mendel now have shown that, so going back here, that I'll repeat the same thing over and over, I hope it'll help, is that here in these plants there must be two determinants, which now we know are alleles, right? In this case they're pure because these are true breeding plants. So each plant has two factors for each character, in this case the color of the flowers. Now the gametes will get only one of this character. Then through fertilization, you reestablish a diploid situation there. So two alleles put together there, two factors there. And so these have the same chance of meeting with each other. And then here, that's the important thing. So then you'll have that when you cross two plants here, they have a mix of them. Now they'll generate gametes, which will have either one form or the other in equal amounts, which have the same chance to meet with each other, given this 3-to-1 ratio. That's the same for all the other characters that he has observed. So now, speaking a bit of genetics here, the way these things are represented in current terms, that's not the way he was doing it. So now, when you are talking about a specific character that has two different possibilities, so two possible alleles, when you talk about the dominant form, you always write it with a capital letter. So purple is dominant over white, so you use P for both of them. P for purple is capital. Small p will refer to as the recessive trait, so it will be white. So it will be white ultimately. And so when you're starting with true breeding plants, obviously these plants will be pure genetically speaking for one or the other. So the purple one will be pure for the purple factors, purple allele. The white one will be pure for the recessive factor there, which is for the white. And then when you have a plant which is a mix, so in this case, when you have pure plants with only one type of allele, you say that they are homozygous. So the starting plants would have been homozygous for the purple allele or homozygous for the white recessive allele. And then when you cross them together, you get plants that they are a mix. They have a mix of these two possible alleles. In this case, you will refer to as heterozygous plants. And what we're talking about here, when you're referring to this description here of the combination of alleles, you're referring to as the genotype. The genotype is the allelic configuration for a specific character. So heterozygous plants are capital P, small p. That's the genotype. That's at the gene level how it is. It might or might not correspond to what we call the phenotype. The phenotype is how that combination of genes will look outside. So the genotype, for example, for the homozygous dominant plants is capital P, for example, for the purple plants. That's the genotype. The phenotype is purple. If you have small p will be white, right? So that's the genotype. White is the phenotype. Now, if you have a combination here, and you can see, so here you have a heterozygous plant, so with a dominant and a recessive form of the same gene, so dominant allele, recessive allele, that's the genotype, heterozygous, that's how you would define the genotype, and the phenotype will be purple. Okay, so now you can see how the way, with the concept of dominance now, Mendel explained how you can have actually different genotypes that ultimately lead to the same phenotype. So the same phenotype is not always corresponding to the same genotype. That's a big explanation that would explain how this happens in many other character studies, not only in plants, but in humans as well. And also it would explain how some characters that disappear in one generation can reappear, like blue eyes, for example. It's often disappear. For example, you have a Caucasian person marrying an Asian person. Usually you tend to have brown color, right? But then it's possible that a few generations later, that blue color that you think is lost, it could actually come back, right? And that's exactly what this explains. Again, summarizing what Mendel's came out with is two things. So that the alleles, so every character is determined by two copies, okay, two alleles, that's how we call them now. And these, one of them is dominant over the other one. And this allele in the formation of the gametes, only one of them ends up in the gametes. Whatever ends up in the gametes is independent from the context. So you have 50% of the gametes with one of the allele, 50% with the other one. And then the way also these gametes combine with each other, so if you have a sperm and an egg combining together, it will be independent from the form that they carry. Okay. So now, let's get into math. Why was Mendel's interpretation so strong? So because what he used is basic statistical data, way of analyzing data. So particularly he used two important rules in statistics, the product rule and the sum rule. So the product rule, going straight to this example here, is how you calculate the possibility of two events happening at the same time. You have an example here. So imagine you have two dice, right? 1 and 2 here. And now you want to calculate the possibility of having a number 4 in both of them. Now what you do, you determine first of all the probability for each dice. So the chance, you have 6 possible numbers in a dice, so for in one dice it's 1 in 6. The same thing for the other one, it's 1 in 6. The possibility of these two events happening at the same time is the product of the two independent events. Okay, so if you have 1 in 6 on the first dice, 1 in 6, the total possibility of having in both of them, it's 1 in 36. Okay, it's like, for example, what is the chance of having these nucleotides randomly happening in a DNA, CAG? If you think, the chance of having a C there, it's 1 in 4. The same thing of having this A in this position is 1 in 4, sorry. And G is 1 in 4. So the total possibility of getting C, A, and G is in the DNA, it's 1 in 4 times 1 in 4 times 1 in 4. So it will be 1 in 4 ultimately to the power of 3. So 16 times 4, blah, blah. You can do the math right there, 1 over 60 something. All right, so you can do that. That's referred to as the product rule. We'll see how this is important to determine the possible genotype in the F2 generation. And the second rule that he used is what is called the sum rule. So now, if you have that different combination can give you the same results over time, the possibility again, it will be the sum of all of them. So here, for example, going back to the example of the dice. Now you want to say, okay, you have 2 dice, you're rolling them. And you want to determine what is the chance of getting a 7 with 2 dice. Now, there are possible combinations that can give you 7 with 2 dice. You can have a 6 in one of them and a 1 in the other one. So that's one possibility here. Then you can have the opposite possibility too, right? The first dice will give you 1, the other one gives you 6. Now you can have that the first dice gives you a 3 and the other one a 4, vice versa. The first one gives you 4, the second one gives you 3. And then you have a 5 and a 2 as a possibility here, okay, vice versa. So yes, 6 possible combinations there. Now the combination, the chance of getting each of these combination is the product of getting the chance one in one dice and this number on the other one. So it's 1 over 6 for each one of them times 1 over 6. Sorry, the pen is really giving me a hard time here. 1 over 6 times, I hope you can follow anyways, times 1 over 6. So 1 in 36. The chance of getting each of these combination is 1 in 36. Now, if you want to figure out, put all these possibilities together, now all you have to do is add them all together, and you see that the overall chance of getting a 7 in your dice is actually 1 over 36 plus 1 over 36. So the combination of all these 6 possible combinations. So 1 in 36 times 6 ultimately, so combination, so 1 in 6. If you don't believe it, I've done this experiment with my daughter. Throw the dice 200 times, you see a similar number overall. So how did he use them? He used them to interpret his data. And so the way you can see this is actually by using what is called a Punnett square. It's a very easy way. When we look at the second crossing, if you remember, so the first filial generation, they were all heterozygous plants, so which would have a dominant form of the allele that gives the color of the flowers and a recessive form, right? And now, when these plants were self-pollinated or cross-pollinated, they gave a ratio of 3:1, right? So how can you represent that combination there is by generating a Punnett square. A Punnett square is just a square that basically allows you to predict when you combine 2 plants or 2 individuals in general that have different possible alleles and different gametes, basically by predicting, it's better if you look at directly at the picture here, predicting the possible combination of gametes you can get. So now, so the Punnett square here you will put, on one side you put the gametes from one of the plants in this case, and here you put the gametes from a second plant. So now each of these plants, if you look, this plant right here could produce a gamete, since you remember one of the rules that Mendel came out with is that each gamete would get only one of the alleles. So the possible combination you can get here is some alleles, some gametes are getting the dominant allele, some others getting the recessive allele. Now, their sorting, if you remember, their segregation is independent from what they carry. So their chance, you should have half of the gametes with the dominant, half with the recessive. Now, the same thing is for this plant. So if you put it here now, 50% would have the dominant, 50% would have the recessive. Okay, so in a Punnett square, you put on one side the gametes from one plant, on the other side the gametes from the other plant. And then now you can calculate the genotype that you get by combining these gametes. So this, what you represent here on the square, is what happens during fertilization. So now you have that if this gamete carrying the dominant gamete from this plant crosses, so gets together with the gamete carrying the dominant allele from the other plant, now you get as a genotype a plant that has both alleles that are dominant, right? By doing that, you can predict, first of all, the genotype and the phenotype too, right? So if they're both dominant, the plant would have the dominant trait. So that would be purple in this case. Not only that, but you can also determine, using the Punnett square, you can determine the genotype that you get and also the genotype and also the probability of getting that genotype, which is nothing else using the product rule, which I just mentioned before. So the product rule is by multiplying the chance of having a gamete with that allele times the chance of having a gamete with this allele right here. So since it's 50-50, now you will get that 1/4 of the plants, 1.5 times 1.5, based on the product rule, would have this type of genotype. Then you can predict for all the other plants here, right? So once you're done, so basically moving through all these steps here, you end up with this final Punnett square here, where you have determined now all the possible genotypes coming from the combination of the different gametes in different plants with their also relative chance, all right? Altogether, obviously, it should give you 1/4 in this case. Now, based on that, you can determine also what will be the phenotype, all right? And so you will come out with 3 out of 4 plants. So it will be, in this case, you would apply the sum rule. So if the chance of getting purple is this plus this will be 3/4 purple and 1/4 white, which is exactly what Mendel observed. Okay, so the Punnett square again here, this is just a way to represent the cross that Mendel did with the F1 generation leading to the F2 generation here, using a more direct way to show it. So you put, again, summarizing, you put the gametes from one plant on one side, on the other one with their relative chance, okay, being there, which is 50-50, and then you determine the genotypes that will you come out from the different crosses. So this cross, and so on here. Based on that, you have determined how many plants would have certain type of genotypes. You can figure out the phenotype associated with that. And therefore, using the sum rule, you can add them all together, coming out with the ratio that you will get in there. Okay? So now, why did I do that? Is so that we can lead to the next step, which is understanding why Mendel could not interpret this data in any different ways. So the first 2 questions I'm going to show you here is why did they come out with 2 alleles? Could he have determined 3 or more alleles? Could he have used them? And you see, he could have not done his interpretation with any other number than 2. And also, why did he come with the conclusion that all these alleles have to segregate in the gametes independently from their own content? Why do you have to have 50-50? Can't you have 60 and 40%? Why? And we'll go through the math right now. So this is the experiment that he did. Again, true breeding plants, I repeat the same thing over and over, I'm sorry, but it gets in. So pure genetic plants here, right? Which would have PNP, then they form the first filial generation, heterozygous, which then gives you a 3-to-1 ratio between the two phenotypes in the second filial generation. Okay, so first thing, there is something left behind there from a previous attempt. Why not 3? Well, you can't go with 3 nor any odd numbers. The reason being that simply you cannot explain how they will be separated in gametes so that you get the same number of alleles in the next generation. So if you have 3 of them, right? So imagine like in this scenario here, these plants have, they're pure true breeding plants, so they must have only one of the alleles, right? If they had 3, that's how they should look, right? All homozygous for that specific. But now you know, so you have two possibilities. Either they send 1.5 into the gametes, they divide into half. Does it make sense? No. Or it's possible that they generate gametes with a single allele in each one of them, right? But now, to reestablish 3 alleles being present in the next generation, then you have to imply that you have 3 gametes fusing together, right? At fertilization, which we know is not happening. Even Mendel knew that. You have only one from a female, one from a male getting together. So you cannot explain how you can reestablish the number of 3 if you use 3 alleles, all right? This is the same for 5 and so on. Now you see also, why couldn't it be 4, 6, 8, and so on? We go through an example of 4 later on. So you can see how this is going to be excluded by the next set of experiment that Mendel did, not the one shown here. Other thing is the independent segregation of these alleles. Why do you have to have 50% of gametes with one specific allele while 50% with the other one? So in Mendel's experiment, so here half of the gametes would have the dominant form and half would have the recessive for both plants, right? Why couldn't it be the other way around? Why could it be that 2/3 of the plants, like in this example, have the dominant and 1/3 have the recessive? Why couldn't that be, right? And again, I want to show you what is the strength of Mendel. So, okay, let's assume this scenario. You could use any number which has now 50-50, okay, if you don't believe me. So, based on this hypothetical wrong scenario, again, now, would you get what Mendel has observed in the F2 generation? Would you meet, so your theory here, would it match with the experimental values of Mendel? Well, they would not. So now we can use the Punnett square, right? So we're making a Punnett square where we put the gametes here produced by one plant and the gametes produced by the other plants. So going back here, they are the same gametes, right? They're both heterozygous here. So, but now what we change here, so the plants here would have some of the gametes here from this plant would have the dominant, some the recessive, but now is not 50-50 anymore. 2/3 would have the dominant and 1/3 would have the recessive form for both plants. Okay, let's assume that there is a preference in the gametes from one of the alleles. And now, so you can calculate the genotype, but also their frequency, their expected frequency, if that was true, right? So if you do your math here, 2/3 times 2/3 is 4/9, right? 2/3 times 1/3 is 2/9, and so on. Now, if you add them all together at the end, you will see that if there was a preference for one allele versus the other in the plants, you would not get the 3-to-1 ratio. Okay, and I went through this slide, I made it up just to show you mathematically that that doesn't work. It works only, you get a 3-to-4 ratio only if these distribute equally between the gametes. Not only that, but there is also no preference between gametes when they have to fuse together during the fertilization, right? So this both assumptions have to be true. So that's the reason why Mendel came out with the conclusion that these alleles have to be segregating independently with the same percentage, same chance in different gametes, okay, and that they will fuse the same way. Now, I hope you buy this, all right? So still could explain this. If you actually do this part here with more than 4, you actually could still get Mendel data for the first type of experiments, okay? However, there are other experiments that Mendel did that exclude a multiple of 2 as alleles. And this is one of them. In this experiment, what Mendel did, and to verify, that's another strength of Mendel, is that he came out with his conclusion, and to confirm them, he did further experiment. One of them was doing different type of crosses. In this case, what he was doing, he was actually crossing the heterozygous plants produced in the previous experiment, not with other heterozygous plants, but with homozygous recessive plants. And the reason why he was doing that is because mathematically he could predict the outcome, and he wanted to see if that was actually correct. So if it's only 2 characters, you can predict that you should get half of the plants purple and half white, ultimately. Okay, you can do it mathematically and predict that. In fact, that's what he saw. Now you can see why 4 alleles would not work. Now, 4 alleles would not work. Let's go through the scenario. So these would have been the first experiment that Mendel did with 4, if you wanted to explain with 4 alleles. That's correct. Okay, but I just wanted to show you why you can use 4 or multiple of 2. So let's assume that now the purple plants have 4 alleles there. Okay, pure, because they are true breeding plants. They have to be pure. So all the gametes they produce should be identical in terms of content. So all the gametes from the purple plant should contain 2 at this point alleles, dominant alleles, and all the plants from the white plant, all the gametes, sorry, from the white plants should be containing a pair of recessive alleles. And now when you cross them together, you get, again, a plant with 4 alleles, that's fine, which would have 2 dominant alleles and 2 recessive one, and will look like the dominant plants. All right. Up to here, everything is good. You see, even if F2, it could be okay. So here, what you have to do, though, is, okay, so you would have an evidence that if plants have 2 dominant alleles and 2 recessive, they acquire the dominant phenotype. Now, this wouldn't give you information on other combination, like this. If you have only one of them, would it be purple or white? And now, you would take this plant, so go into the second step. So let's see what you would expect to get there. So now, if you look at the gametes here that could be produced, now all the gametes here could give, sorry, I forgot one thing. So if you do the next step here, so you could have 6 possible combination here. You could get 1/6 of the gametes with a dominant form, both of them, 4/6 with a mix of them, and 1/6. You can do the possible combination there. You have 6 possible combination here, and with the recessive form 1/6 in the end there. Okay, if you mix them together, now you assume that these still give you white, then you still come out with a 3-to-4 ratio. However, now if you do the second experiment based on this theory, okay, so where you're crossing a plant like this, so if you assume that this gives you white, and you have to assume it to justify the results of the first experiment, you can do a Punnett square with this value. You'll see that if P, capital P, and 3 small p give you white, then you get a 3-4-1 ratio. If you get it that this should give you purple, then you wouldn't get it. But assume that this gives you white. Okay, this would explain the first experiment. But now this doesn't explain the next experiment, which is if you cross this with one of these plants, right? So that's the experiment, the next experiment that Mendel did. So if you cross these plants together, now that's what you should get. So this plant here gives you only gametes that contain 2 copies of the recessive allele, whereas these plants here can give you these possible combinations here, which are on this side. So these are coming from the purple plant, and these are coming from the white plant, assuming that there are 4 alleles. Again, I'm trying to destroy the possibility that there are 4 alleles. And now they are the possible combination. So you should get some plants having 2 dominant and 2 recessive alleles, some plants having 1 dominant and 3 recessive alleles, and some plants having all recessive alleles. Now, if you do this, now we know that this should be purple, right? You have to make this assumption because these are purple, right? Now, this one we have determined here from the first experiment, which I didn't show you, you would have to accept that this gives you white. Okay, if this gives you white, now you would have the calculated sum. So 1/6 of the plants will be purple in a cross like that, and 5/6 will be white, which is not what he saw. He saw half plants being purple and half being white. That's the experimental value. So meaning that the theory based on 4 alleles doesn't support that. Even if this was purple, you set this as purple, now you would have 5/6 being purple and 1/6 being white. Still, it's no 1-to-1. It's not 50-50, which is what he had observed in this second experiment. What he saw is that 50% of the plants were purple and were white. So that's the reason why the data from Mendel can only be explained by 2 alleles that do segregate independently from their content in the gametes, okay, giving you a 50% chance of the gametes having one or the other. And even when they fuse, one gamete has a 50% chance of meeting the other gametes independently from the content, okay, giving you those ratios 3-to-4 and 1-to-1 in the second type. So that was actually the plants. Anyways, skipping that slide there. Now Mendel went ahead and did again strengthened his theory by doing other experiments. The other experiments that he did, it was what is referred to as dihybrid cross, where instead of working with a single character, so just working with purple, for example, he actually put together 2 different characters. So for example, here it tested the color and the shape of the seeds together at the same time, and starting from pure plants. So in this case, it started from plants that had pure round and yellow seeds. So the shape and the color of the seeds are the 2 characters, and other crossing them with pure plants, true breeding plants that had wrinkled and green seeds. So here too, you only have one possible combination of gametes produced there, which will produce heterozygous plants for both characters at this point. And then what he did, exactly in the first experiment, took these plants and let them self-pollinate or cross-pollinate. So these plants could only pollinate with each other. And he predicted the numbers, which indeed were supporting what he measured. All right, based on his theory of the 2 alleles, he could actually come out with a ratio, theoretical ratio 9-to-3 to 3-to-1, which will be, this is the theoretical one you will come out, presented as a Punnett square. Again, where you put all the possible combination of gametes here from one plant, matching the one from the other plant here. And by doing that, you get the possible genotype produced with their also expected frequency in the F2 generation, and there from there you can calculate also the expected frequency of the different phenotypes. And so what he came out with, it was that 9 out of 16 possible plants should have round peas and yellow peas. 3 out of 16 should have wrinkled and yellow. 3 out of 16 should be round and green, and only 1 out of 16 should have the recessive phenotypes for both characters, which are wrinkled and green. All right. Now, these data were the beginning of genetics. Okay, so it was the first time that someone explained how characters can be transferred by 2 copies, okay, from one generation to the other one. One thing important to understand is that these represent only one way, now we know, in which characters can be transferred. There are many other factors that do add to them. There is epigenetics, there is different ways. There's sometimes you have more than 2 possible traits for a character. Sometimes there is no dominance in place there. But Mendel was very lucky because all of the characters that he studied, first of all, they were all monogenetics. So there was only one gene determining the color of the plants, the color of the flowers. Only one gene determining the color of the seeds or the shape of the seeds. So if it had more than that, his number would have not matched his theory. Other thing, there was always dominance there. No matter what characters was working, they were all determined by two possible alleles, and one of them was always dominant over the other. It's not always true. We'll see some example in a second. The other thing is that all of these genes were in different chromosomes, or they were in the same chromosome but very, very far away. If they had been in the same chromosome, very close to each other, he would have not got the ratio that he had. Usually we have another lecture after this. I'm not going to talk about this, but trust me, he was a very lucky man overall as well. Other than being an extremely smart man, he was also very lucky. Now, we talked about Mendel's experiment. His conclusion was about factors. Okay, he didn't know what these factors were. He didn't know that they were in the DNA and so on. The scientist that actually extended his theory to the chromosomes was Sutton. This is a scientist that was studying actually the chromosomes, and he could see that chromosomes were coming mostly in pairs, right, in most organisms. And so he just made the bridge. He bridged the conclusion by Mendel to the cytogenetic conclusion that he had. And so he hypothesized that these alleles, these factors were actually in the chromosomes, right? And he was doing that because he observed the chromosome come in pairs, exactly like these factors. And then also that they do separate during meiosis, you know that, independently from each other, right? You have one chromosome going on one side and one gamete, one on the other one, which matches what Mendel was saying. And then also that this is, in the end, they're brought together through fertilization, one from the male, one from the female. So there were a lot of similarities between the behavior of the chromosomes and the behavior of this factor that Mendel had hypothesized. And so he came out with what is called the chromosome theory of inheritance. It basically says that these factors that Mendel was talking about are indeed in chromosomes. They're located. And now we know it's true. Now we know that there are genes are what determine what carry these factors, and they exist in different variables, and they are located in chromosomes, and they do split exactly as Mendel had described. And so just expanding a few words here. So we know now it's gene carrying them. Genes are always found, so specific genes are always found in specific location in specific chromosome. For example, imagine this being chromosome 7. You have one copy from your father, one from your mother. This is a homologous pair of chromosomes. You remember, they are very similar but not identical, differently from sister chromatids, as we say to it. And the difference is exactly in the content of the genes. So now you would have that genes are found always in the same location in a specific chromosome. Imagine this is the gene for the color of the flowers. It would always be present in the same chromosome in the plant, in the same location. And this is referred to as the locus. This is the locus where the gene is. Okay. And now there are different variables there referred to as alleles. So for example, this will be the dominant one for the color of the plants, will be the flowers will be purple, and this will be white. So the gene for the color of the flowers is always located in the same chromosomes, right, in the specific locus, and each of the two chromosomes could have different variables of the same gene referred to as alleles. Okay, and then they separate again in the segregate independently, exactly as chromosomes do during meiosis, and so on. Now